So we use the online software called Ensemble. And then here, you can select different species. Here we will choose zebrafish as our target species. And then you can you can type in the gene of interest you want to check. In this case, we will check toll-like receptor 3. And then you enter this website, select the species, and enter the target gene name, and then click Go. Then you will see here, there are several icons, and then first one will be your gene, the gene of interest that you are looking for. And then you click the result displayed in the red box. Here also, you click transcript ID. If there are more than one, usually the first one can work. And then after you click it, you will get the information above, like above. You will see how many introns and how many exons for this gene. And in this way, the white box represents the untranslated region, which we will not design our primer on it. And the brown box represents the exons, and the line represents the introns. Then, so follow the rule, we will design primers on exon-exon junctions. So we will click exons. We are trying to get the exon, and then it will jump to another web page that shows you the sequence. Here you can see there are several sequences, and then in between you have lowercase of the ATCG, which represent the introns. So you will remember not to design the sequence on intron, but only on exon and exon junctions. So you locate the exon-exon junction here. Then you copy the required sequence to Word program for operation. You select the primer according to the key points explained above. First, select and paste the 20 nucleotide, and then paste it in oligonucleotide properties calculator. Or you can use other software, online software, that can do the same thing. Then, according to the TM value requirement for forward primer and reverse primer, nucleotides should be added or deleted appropriately. Finally, remember to perform reverse complement on the reverse primer, and then complete the primer design. Then you can see here, for using this oligo property calculator, underneath you can see that salt adjusted TM value for the primer is 60.1. That is the TM value you want to compare for forward and reverse primer. Then we will use zebrafish toll-like receptor 3 as an example, and we will design primer as follows. You take exon 4 and exon 5. In between, you will delete the intron sequence. And then you will design your forward primer. We say that we have roughly 20 to 25 nucleotides. First, you will have maybe 18 to 20 nucleotides on the forward primer on exon 4, followed by 5 nucleotides on exon 5. So this sequence, GATCC ATG to TTCA on exon 5 will become together to be your forward primer. And then you will calculate roughly 100 base pair, and then design reverse primer for the zebrafish TL3. So the reverse primer will be TGGAAGA until CAACC. And here, remember to do reverse complement for them. In this case, the PCR product size is 109 base pair. All follow the rules. So let's just get back to the rules and just remind you some of the main points of the primer design. First, your primer should be designed close to the stop codon, as close as possible, due to the 3' bias when using oligo-DT. What is 3' bias? We mentioned before, when we are doing the reverse transcription using oligo-DT as primer, because they contain many T in the primer, which can complementary binding to the poly-A tail of the messenger RNA. So the oligo-DT will anneal to the poly-A tail and then start the reverse transcription using the reverse transcriptase. And then the reverse transcriptase will start making cDNA from the end to the head. So we'll start from the 3' area until the 5' area. So imagine that sometimes the enzyme will stop doing their work because they are too tired or something. So it will stop in the center. If you design your primer in the beginning of the sequence, then maybe that sequence, the mRNA sequence, was not properly reverse transcribed by the reverse transcriptase, so that you cannot detect anything. That's why we ask you, try to consider to design your primer as close to stop codon as possible. Next, primers should cross exon-exon junctions. We just talked about 4 different scenarios. Please, if you want to check again, you can go back to the previous slides and have a look. There is only one exon, or at least one exon is too long, then no need to apply. Which means that if you are going to design a primer set on an intronless gene, which means that the gene does not have any intron, only have exon, then you cannot apply your primer on cross exon-exon junction. The forward primer will have 15 nucleotides on one of the exon, for example exon 1, and then you will have another 5 nucleotides on exon 2. And then the reverse primer will have the same thing, 5 nucleotides on exon 2 and then 15 nucleotides on exon 3. Once these exons are being merged together and the intron was removed in the mature messenger RNA that is transcribed, reverse transcribed into your cDNA, your exon 1 and exon 2 and exon 3 will be linked together. In this case, only the primers that you design cross exon-exon can properly binding to the messenger RNA or binding to the cDNA that you generated from messenger RNA, but will not binding to your corresponding genomic DNA, which contains intron to intervene the combine of the primer to exon 1 partially and the exon 2 partially. Primer should be between roughly 18 to 25 nucleotide long. You can set it as 20 nucleotide in the beginning and adjust it accordingly to the temperature. You can use the calculator like oligonucleotide properties calculator to know their melting temperature. The temperature must be between 57 to 62, preferably around 60 degrees. The Tm value difference between forward and reverse primers should not be greater than 2 Celsius degrees. The size of PCR product is optimized to be 50 to 200 base pair, but I would rather recommend to roughly around 100 base pair. Thank you for listening. I'm Lipo Chang.